mince pie.
Yeah,
Oh, well done, well done.
And Christmas cakes as well.
Fred?
I couldn't face the mince pie.
Sue.
Yeah?
Sue, who was that? Who cooked those amazing mince pies at the swim?
Oh, my friend Ella.
Did you make them yourself, Sue?
I don't cook, you know, Bill.
Oh, didn't you not?
I don't just be cooking.
Oh, I see the box there, co-op.
White Rose, actually.
Yeah, right.
Vicky, are you sitting in your garden?
I am. It's lovely, isn't it?
It's very pretty garden you've got.
Oh, Rosemary, you're there. Hi, Rosemary. It's Judith. Hi.
Oh, hello, Rosemary. Hi.
Oh, I just sent you a message to see if you were okay getting on Zoom, but you've done it. Brilliant.
I've got to put myself on mute because my dog's been
Oh,
Can you
nice
hear
to see you anyway.
Yeah.
Oh, hi, Rosemary.
Cheers.
Hello,
Fred, it's lovely to see you. We seem to, I've lost Professor Leigh.
Yes, I seem to have lost him and Caroline.
He obviously didn't like what he saw.
Had it yet?
Oh, there he is. He's back. He's got a hat.
Oh, there, yes, now I can see him. There he is. Yes, hiding away.
I'm here as well.
Taking my bubbles.
Okay.
Everyone keeps jumping around the screens.
Oh, wow.
I take my bubbles off now.
Yeah.
And Maggie, nice hat.
A Christmas tree hat.
We need to mute everyone.
Yeah, we'll have to mute
always
everything.
doesn't be saying what you always declare. Do it when you're doing it.
That's okay.
Doesn't matter what you want to do it.
I'm loving it.
That's amazing.
It's always lovely to have a little girl telling us she needs to run the meeting.
Hi, Francis.
Are there more people waiting to come in, Dean?
Not yet, no. I mean, it's 46, I think, at the moment.
Wow.
26.
We need a bigger screen.
They
We'll have to all breathe in and squash up.
Oh, wow.
Oh, here we go. Somebody else here.
Wow.
I shan't stay. I shall just wish you all. I shall just wish you all a very happy Christmas from me and from Snowy. And I love you all.
Oh, happy Christmas.
Happy Christmas.
Recorded, Chris, so we can send the link.
Raise a glass to you. Happy Christmas.
Happy Christmas.
The lady sit on the bloke's laps. Will it make the picture smaller?
Yes.
It's funny. I don't know why.
Has somebody disappeared? Has Anne gone? Or have I not got everybody on the screen?
You can try it first, Bill.
Sounded like Emma.
Hey, Barnes, I'll just something for you there.
Just go.
No, she doesn't want to sit on my lap.
It's very confusing.
Amazing. Two full screens. Wow.
Yeah. Wow.
Brilliant, isn't it? How do you do the second? I've only got one full screen. Am I missing something?
Yeah,
A trick?
click on the side.
Click on the side.
Right.
You better scroll
You
along.
the other.
Yeah, or you should get an arrow on the side that you can switch between.
There you are, Lee.
All
I
the
needed
other
you.
ones coming in.
Lee, I needed you to tell me that. I found the arrow. Yeah.
Yeah. Find all those other people.
Brilliant. Wow.
Oh, I'm upside down.
What do we do if we have more than
Who's upside down?
Me.
Is she from Australia, Bev? Are you in Australia? She's gone down under for the Christmas.
Might be a bit warmer down there.
Is it warmer down there?
I guess. It's their summer. It's their summer, I think.
Yeah.
I've just seen you, Steve. Hello, Steve. Colin's son. Hi.
Hi, lovely to see you. Hi.
Hi, Jean.
Hello.
Hi, Jean. Hi, Matt.
you.
I've just seen
Can someone
Dawn.
help me? I've gone to two screens and now I can't get back
Taking you off.
I don't have a clue.
Have you
Look
got a,
on the side, Maureen.
yeah, have you got,
You should have a little arrow on the side.
I've got the arrow, but I'm pressing it, but it's saying
are you doing it? So do the left-hand one to, or the right, oh, sorry.
oh,
There's a left and a right hand.
Hello,
Yes.
Francis. I see Francis.
Hi.
Well,
Hi,
we muted.
Francis.
I can see Pip now.
I can see Dawn,
Hi, Chris.
me and Jean,
Hey, hey, hey.
Oh, it's all right.
Barnaby. I'm not sure who
Thanks,
Barnaby
your
is.
volume.
Barnaby's the friend of ours.
Oh, hello.
I just dropped the arrow.
to me, so they all know each other.
Where's everyone gone? While I screen?
Well, I just thought it was the southern area, and it would be lots of people. Oh, okay.
Right.
You're in charge of the television. No.
It's called organised chaos.
It's
not very organised. Fine.
Yes.
Obviously, we run the
Not everyone will want to have their video on, will they?
Right, I think. Maureen, do you want to start?
Sorry, Dean.
Would you like to start?
Yes. All I can say is welcome to everybody. This is absolutely amazing. I can honestly say that I don't know everybody, and I'm looking at all the screens and I don't know everybody. So I suppose I better introduce myself. I'm Maureen O'Neill, and I chair the West Sussex South branch.
I'm just going to mute everybody. Hold on 2 seconds.
Oh.
And I'll open Maureen up to mute Maureen. Maureen, where have you gone?
Muted.
There you go. You can hear you now, Maureen.
Was I muted?
You were. Sorry.
Sorry. All I can say is it's absolutely amazing. I've never seen so many people come together. This is wonderful. And I can hear people saying, does everybody know everybody? I don't know everybody. But all I can say, this is incredible to have you all here. If you don't know who I am, I'm Maureen O'Neill, and I'm chair of the West Sussex South branch. We're going to try, there's so many people here, it's going to be such good fun. We're going to sort of pull you through the next hour and 15 minutes, we think is probably what it's going to take. But what I'd really like to say is that we're going through some very strange times. But it's really heartwarming to know that we can still gather together, even though it might be remotely. And actually, I quite like it. It's good fun. It's really, really wonderful to have so many people here. I hope you all enjoy the evening. And I'm hoping that you've all got a candle, a glass of wine. Cheers. And a mince pie, which I haven't got, but there we go. Think and extend our thanks to Professor Nigel Leigh from Brighton and Sussex Medical School and Dr. Caroline Vance from King's College London for joining us tonight. And also a really special thank you to Frankie for her performance tonight to accompany our memory wall. Frankie, you're a star. Thank you very much. All I can say is that running a branch involves so many fantastic members and supporters. And West Sussex South is very lucky to have all of those people. We have a great committee. We have fantastic association visitors. And I'd really like to thank them all for their help and support throughout this year, which has been a very, very difficult year. I'd also like to reassure everyone that Angela and Sue Patty, Valerie and Prue are all in contact with our people with MND and look forward to when they can resume their face-to-face visits. Everybody has pulled together through what has been a difficult year, ensuring the branch can go from strength to strength. This has been led by our new subcommittee members, and special thanks must go to Dean. You are a star. He's re-energized us all with his enthusiasm and, most importantly, his understanding of technology. He has enabled us to reach out, promote awareness, fundraise, and support people in a way that we haven't been able to do before, and has been needed in so many occasions by so many people on the committee to help and support. You need to go on holiday, Dean, somewhere like Mauritius at some point, babes. Dean and Nikki have organised this evening, and it's taken a lot to get this together. I'd like to thank you so much for making this happen. You are amazing, and I don't think any of us can thank you enough for helping us out, because we wouldn't be able to do this. We wanted not to go on for too long because we've got so much to fit in tonight. So we're going to have a sort of short year-end review with some pictures of things that have gone on this year, so that you all realise that we are still here and we're all still doing things to support all of those people that have MND. Dean?
Yeah. Mm-hmm.
Branch fundraising has actually been really successful over the last 12 months. Sorry, I just need, no, it's fine. Even with the problems that COVID has put our way, although it's been a really tough year for everybody in every way, we've still managed through fundraising. Are the pictures going up, Dean?
Yeah.
Although it's been a really tough year, we've still managed through fundraising, donations and legacies to raise £55,000. We continue to appreciate all of our members and their families' enthusiasm and all of the efforts to raise money for the branch. This enables us to help people with MND in our area. There are so many people we'd like to thank for supporting us throughout 2020. We'd like to thank Lynn Crompton's family for their kind donations to the branch, especially John and Barbara Alban and all of their friends and family who support us continually. The Body Shop for support with our bingo night. They are still supporting us through 2021. Everybody involved and who generously donated to the Sandy Barrett Tribute Golf Day. It was a fabulous day. Sue Acaster, the Worthing Running Club with the Mission 5000. John Zimmer, who walked the South Downs Way with his friends and their dogs. Steve Maple and Tesco's at Holmbush for their kind donation. And especially Nikki O'Neill, our amazing secretary, and her family and friends for her swim in the sea. You are mad, Nikki, but well done. Thank you very much. Sue and her friends will be running on Saturday the 12th along Worthing Seafront dressed in Santa costumes. It would be great if you could support this event. You can find loads more information about events on our website. We already have some really exciting future fundraising events in 2021 and look forward to seeing some of you there. So I'd just like to thank everybody. You've been amazing. We are so lucky. I'd now really like to introduce Professor Leigh.
We're going to do the video first.
Which video?
The memorial.
Sorry, Sorry, yes.
So now will be a good time. We're going to light the candles and we're going to have a video that Frankie's prepared of the memory wall. And we'll do that before the Professor Leigh comes to speak. So if you've got your candles ready, we can try and start to light them now without trying to burn the place down.
I was just about to say, be very, very careful.
There we go. Actually got a real candle.
Yes.
Okay, let me see if I can get this.
Holy night, the stars are brightly shining. It is the night of our dear Saviour's birth. Long lay the world in sin and error pining, till he appeared and his soul felt its worth. A thrill of hope, the weary world rejoices, for yonder breaks a new and glorious morn. Fall on your knees, oh, hear the angel voices, oh, night divine, all night when Christ was born, all night divine, all night divine. Truly he taught to love one another. His law is love, and his gospel is peace. Chains shall he break, for the slave is our brother, and in his name all oppression shall cease. Sweet hymns of joy in grateful chorus raise we, yet all within us praise his holy name. Fall on your knees, oh, hear the angel voices, oh, night divine, oh, night when Christ was born, oh, night divine, all night divine.
Thank you so much, Frankie. That was beautiful. Thank you. I think think it would be really nice now to introduce you to Professor Leigh, who will be giving us some messages of hope as he outlines developments in the latest drug trials. There will be some time for questions later if anybody has any. So I think it's best just to hand over to Professor Leigh.
Yes, let's see if I find him. There he is.
Is that my cue then?
That is, yes.
Listen, thank you all. What a fantastically impressive group you are. What achievements you've made. I think what I was going to do is be very brief and really leave it open for questions, because and also make sure I leave time for Caroline. Hello, Caroline. I haven't seen Caroline for a long time, but we were colleagues together at King's for a while. So please shut me up, Dean, whenever you feel appropriate, and make sure I try not to hog the time. And also, as I said, try and give plenty of time. So my opening message really is quite brief. It's why do clinical researchers, that's people who are seeing people with MND, ALS, I use the terms interchangeably. If I lapse from one to the other, there's no significance there. Why do we clinicians feel that this is genuinely a time of enhanced and real hope, when, to be honest, and you all know as well as I do, that we don't have in hand a cure or even a drug that we can hand out, which tomorrow will arrest this pernicious condition in its tracks. That's what we are searching for, and that's what we don't have. But why is there so much hope? Well, I think there are various conditions which have come together, which really altered the picture, I think, for me. And I've been in the field, I don't know, a lot, probably, well, Caroline was probably in her infancy when I started. I will let her comment on that. So I've been around for all too long, and we've failed you all in that time. Now, as you can appreciate, it's not through want of trying. We've done dozens of clinical trials at King's, when I was at King's for many years, and even over the last 10 years at Brighton. And yet, what have we got to show you for it? Well, we have riluzole, which some of you will know. It doesn't make anybody feel any better, and it doesn't make you aware that there's an impact on the disease. You have to believe in statistics. You have to believe, I'm one of the lucky ones. It has made a difference to me, but you certainly can't sense it. So why is it different? Well, I think what's happened, and Caroline may well comment from a slightly different vantage point, I think never before have we reached such a joined up approach to research, and that means from the laboratory at the level, from a single molecule, if you like, or single molecules, to cells, to animals, which are still part of the research team, if you like, through to the bedside. Never have we been more joined up. The reason for that, and Caroline will probably go into that, is the genetic discoveries. I'm not going to dwell on that because I think Caroline may well has more authority on that than I do. But we've discovered so much, including Caroline's discoveries over the last 10 years, that we're beginning to see that the causation may be beginning, the jigsaw of causation may be beginning to come together. We now know that MND is not just a single disease. I always tell my patients that it's as many diseases as there are individuals with MND. That's a bit scary, because how do we attack that? Well, that's a big question. You can grill me on that if you wish. But what we've done is to say, listen, the approach that we've had of picking off one company who's got their pet drug and saying, let's test this, or some academic like me, well, I've got a pet notion, let's test this or that compound. We said, let's try and bring together a more joined up approach. And this we've seen in what we call platforms, which are just beginning to be rolled out. Now, some of you will know of the MND SMART initiative. Is that true? I think most people will have come across that. So that's a platform. There are several trials in one. That's being run from Edinburgh, but it's going to come to Brighton very soon. Directly, we're allowed to open up again and around the country. There's a big platform that is a lot of people collaborating nationally and internationally for a collaboration called TRICALS. Just trying to remember what TRICALS stands for. It doesn't really matter. It's something to do with ALS. Caroline might remember. Amar Al-Chalabi at King's is one of the leading proponents. And again, that will roll out a series of trials so that people can move into a trial. And if that one seems to be failing at an early stage, they can move into it, and so on and so forth. This is what's happening in MS, for example, and Parkinson's as well. And then, of course, there's the trial close to my heart, which is the one I'm running with my colleagues in Paris and Nîmes in France, which is the Miracles trial. And John Alban was very much a part of that, and others maybe. And John was a hero. So the heroes of these stories are not the doctors or nurses running them. The heroes for me are the people who say, okay, I will join this thing. I will put up with blood tests. I will put up with lumbar punctures, particularly lumbar punctures done by a semi-pensioned professor of neurology. That's a pretty scary thing. John was very brave that way. And so these are all collaborations, as I said, of many centers. And what they do is they don't just test a drug in a group of patients. They also look beyond that to say, why, if it's not working, why didn't it work? Are we any the wiser at the end of this? And also, they're integrating, you can hear my dog barking in the background. Sorry, I can't muzzle him. That's Charlie, sends his greetings. But we have scientists working in labs in all these projects, taking your bloods and finding ways to understand more about the disease, as well as finding perhaps a therapy. So that's really my very positive message. Now, when will we deliver something? Of course, I can't tell you that. But I think never have the chances for a much more speedy delivery. And these things do take time for good reason. I mean, good reason for us, not good reason for people at the sharp end, but there are scientific reasons why we can't speed it up too fast, because it's a very complicated condition, etc. But we want to do it faster than we do it at present, and never has the time been better for that. Now, I think I've probably said enough to give you that background as to why I think we're really accelerating the process. And I'm going to stop and just ask people to ask questions. I can tell you more about, for example, the Miracles trial. I think that may not be the purpose, but I'm in your hands.
Anybody that wants to ask a question, please just put their finger up and we can go straight to you.
I'll have to rely on you to watch. If Dean or you, Maureen, are monitoring that, then I'll leave it up to you.
I have a question. Is funding a problem?
Well, it is, of course, and what you're doing to raise money towards research is absolutely vital. We cannot do it without substantial money. And these things are expensive. I'll tell you how much Miracles cost. And that's only looking at 220 patients in UK and France in 17 MND centers. It costs 6 million euros straight off. And we got about another 3 million euros that we've gone out to get to amplify because we didn't have enough. That was the cutoff. That's all there was on offer. Companies may spend 50 million, 100 million on a trial. So this is an academic-led trial, which has the freedom to operate which you don't always get in the company-led trials. And it means we can integrate much more science in it. And we do it, I suppose, on a shoestring. But, you know, obviously we have enough money we have to have enough money to do what we deliver. But yes, I mean, money is an issue. And the other issue is, I have to say, is international collaboration, because not everybody will fund that. Only the European Union would have funded what we did. Nobody else. The MRC, the Wellcome Trust, NIHR wouldn't do that. And so that's a worry for us in the future.
Leigh, do you want to
I think international collaboration is the lifeblood of the whole thing.
Leigh, do you want to ask a question?
Yes. Okay, I'm asking on behalf of Leigh, so forgive me if I get this wrong, Leigh. Leigh would like to know, when will the Miracle study be able to report its findings?
Yes, well, I can tell you. So the trial should be finished by the middle of next year. The last patient out should be about July. It depends then on how quickly, of course, COVID has not made life easy for that. We could check and double check the data. You've got to have accurate data. That will take us a couple of months anyway, depending on how much we nag our colleagues, and we will nag them a lot, I can tell you. And we hope we will certainly have the answer before the end of the year. And the answer is as quickly as we can after the end of the trial. But within, if we could within days, it's more likely to be a couple of months because we have to check the data.
Okay, thank you.
Unless our independent data monitoring committee says, we recommend you finish it earlier, either because it's not safe or because it's so effective that it would be unethical to do.
Yeah. Can you say something else?
No, sometime in 2021.
Sometime in 2021. Yeah. Thank you.
Has anybody else got questions? Dean, I can't see everybody, but anybody else?
Yes.
I have another question if no one else has.
Yeah,
Sorry.
Yeah, there is a guy, I've got it just known as Denise. He's got his hand up. If you could unmute yourself. If you can unmute yourself.
I am unmuted, I think.
There you go.
Okay, I'm an interloper actually from up here in Norfolk.
Lovely.
I hope you don't mind me interloping. Anyway,
no, well,
I was diagnosed with MND 7 years ago. Now, I've got a fellow up my street who's always into drugs, not that sort of drugs. He's recommending that I should try something called leucine, Leucine. Now, I've read up a bit on that, but understand not too much. Does Mr. Leigh know anything about this drug?
Well, it's an amino acid. It's part of our natural diet. It's in meat, you know, in proteins. There's no convincing evidence, I have to say, scientifically. So there's no, I can't make a recommendation based on any science. So it's, if you like, a complementary medicine for which there's no standard, if you like, scientific data. So all I can say is that it shouldn't be in any way harmful, but there's no evidence of its benefit.
Yeah, when you read into it, it looks like it regenerates your muscles.
Well, there's much talk about that. As I say, we just don't have any hard evidence on it, really.
So am I going to have any problems if I try it?
Listen, I can't answer a clinical question.
Okay.
Because I can't mislead you. So, and the GMC would be on my back if I started.
Okay, that's fair enough.
But I do sympathize with you. I mean, it's a natural compound, but you need to really consult your own doctors, etc. I can't really answer that.
Well, we know the testing you're coming down here to Bury St Edmunds, which of course is in Suffolk, near to us, because I'd logged in to be a guinea pig in Edinburgh, and they referred us to, it's going to start in Cambridge, but now we understand Bury St Edmunds is picking it up, and that's 20 odd miles from me.
That'll be good. Yeah, well, I was just talking to them today by Zoom, the Edinburgh people, and they're trying to get it set up all around the country. And of course, directly we're allowed to by our hospitals. At the moment, everything's on hold because of COVID.
Yeah, absolutely.
I think it's an exciting project.
There's another couple of questions coming in. So Kate's got her hand up.
Oh, hello, everybody. Professor Leigh, will Brexit affect your collaboration with partners at all?
Well, thank you. I hope not. I mean, there is uncertainty, of course. Luckily, because we're already funded, there's no problem there. As we checked that, of course, with the European Commission. I think the uncertainty lies with future studies. And also, we do have a bit of a worry because the drug it's a natural compound again called interleukin-2. We have to manufacture it, if you like, put it together for injection in the UK and send it out to the 10 centers in France. We hope that it won't be impounded because it goes off very quickly. So, you know, that's a slight worry. That won't affect people in the UK because we make it in the UK, but it could affect the centers in France. And what we don't want to do is to upset the security of the data. We don't want to damage the quality of the data. So that's a worry, a slight worry, but I think it's going to be all right.
Eric, you had a question.
Who's this?
Another question, Erica, and
Hi there, Professor Leigh. Question regarding, are you collaborating with anybody outside of Europe, such as the USA or in China?
Yes, well, not personally in China at the moment, but we do collaborate and have done over many years with many colleagues in the States. It varies on the project. It depends on the science. So if you've got somebody in the States who's got know-how and knowledge and skills that you haven't got, and vice versa, it's natural to collaborate. And you know, of course, these days how easy it is to communicate with people without necessarily having to visit San Francisco or someone desirable like that, or somewhere less desirable. At the moment, our main collaborations happen to be in Europe, but you know, I've run many trials with Europe as an equal partner with USA and Canada. Yeah, China is very interesting, and certainly people are increasingly collaborating there. Good science.
Well, sorry, someone else's hand go up there.
Mine was a similar question in that, is funding greater in America?
Yes, big time.
But
At
they
least
have
10 times, I would say. I don't know, I haven't got the figures from all the, because there are several groups in America. There's the ALS Association, there's the Muscular Dystrophy Association, there's various other charities. I wouldn't be surprised if it's you know, equivalent of $100 million and so on every year, but I can't quote you that. It's going to be big.
But still no breakthroughs.
You can't buy breakthroughs with money. That's the problem. You can buy the time and the researchers, the talent, and that's, you know, you won't get anywhere if you don't have talented researchers. I mean, the young ones that we're bringing on. And luckily, we, like Caroline, you know, we have some really world-class researchers here and in Europe and in the States, because, as I said, the doors are opening for ALS research. There are dozens of ideas out there because of the discoveries in genetics and cell biology and the collaborative atmosphere. So I think never has there been a time where it's more attractive for a young, ambitious scientist or clinician to work on MND.
Graham, I
think is a
hand up there as well.
Sorry?
Graham has his hand up there. Hello, Graham. Do you want to come off on mute?
I am. Hello. You hear me?
Yeah.
I just wondered, why does everything need to stop because of COVID? I don't really understand why the clinical trials can't still continue.
Well, what actually happened was that first there was the concern about transmission, you know, and everybody went into lockdown, and the hospitals diverted because they had to. They had no choice, I think. Nobody knew what was going to happen. They diverted their resources, including the clinical researchers, into looking after COVID you know, that was in the first wave, etc. And then there were the continuing concerns about patients coming up, you know, just transmission within hospitals.
I mean,
And so it was a safety issue, as well as the fact that the trials on COVID vaccines took over big time, as you can imagine. So yes, I mean, I think, well, I mean, we haven't stopped doing the trials. So we switched to distance
you said the same thing down there.
because we were not going to lose track of miracles. You know, never were we going to let that stop. So we were lucky that we just managed to get, we had to close down just when we'd finished recruitment and got all our blood and spinal fluid samples pretty much all. So we were lucky. We could then talk to patients and do videos. I've been seeing patients again since June. So, you know, we just have to adapt, and we have. So yes, it's not been easy, and some trials have had to stop, but it will get going again in the new year, I think. Let's hope that COVID, you know, is going to ease off, but we don't know. I don't know if that answers your question. It's a combination of factors.
Following on from that, just following on from that, the fact that they now seem to have a vaccine for COVID within 10 months when it normally takes 10 years, do you think that there will be any knock-on effect to other treatments, you know, and discoveries from that?
Yeah, I would like to think so, and I'm sure we should try hard to learn any lessons. That is about adapting and not being quite so rigid about how we do trials, because of course we have to do them according to what's called regulatory protocols, because if we don't, nobody will believe it at the end, or we won't license if it worked. And that doesn't do anybody any good, because ultimately we've got to get it out to people with the condition. But I think distance assessments can be done depending on different trials. You can't do a lumbar puncture at distance yet. If you need that, most trials don't. You've got to come to close proximity. The problem, I think, with MND or Alzheimer's or other neurological conditions is the brain and the nervous system are so complex that a virus, you know, they knew right at the beginning, within 2 weeks, they knew the structure of that virus and they knew exactly what to do to target it with a vaccine. We're dealing with a black box still. We have flashes of light in that black box all the time, but we still don't really understand how the system of degeneration works. And, you know, we're working on that all the time, and Caroline may, I want to hear what she says, but we're still a long way from really saying, oh, that's the target. Now, that's what I would like to see in my professional life. I would like, the light bulb moment when you realize how it all comes together and you realize that what you've been chasing is that, not all those other things. And that could happen at any day, honestly, but it hasn't happened yet.
Thank you.
Nigel, thank you so much. That's been fantastic. I think this is a really good point, probably, to bring in Caroline, if we could. So I'd like to introduce Dr. Caroline Vance, and one of our new young researchers, which is really exciting. So hello, Caroline, welcome. Thank you very much for coming to talk to us.
Thank you very much.
And there probably will be some questions after, if you don't mind.
No, that's absolutely fine. I like the idea of being described young. I'll take that.
You're younger than I am.
Yeah, well, at least.
So, thank you very much for inviting me. Thank you for all your fundraising. I wanted to give a little bit of background about me. Nigel remembers me. Does he remember that he was on my interview panel for my PhD way back when?
Was I really? No, I didn't.
Yeah. So
well, how right I was.
But we were at King's. So I did my PhD with somebody called Professor Chris Shaw at King's College London, and my interest was originally in the genetics of motor neuron disease. So I work on, and I still work on, the inherited forms of the disease. on the basis that from those we can go on and learn more about the sporadic forms of the disease, because they give us our really big clues and keys as to what is happening in disease. And I know that at times research can seem frustrating, you know, but for example, just, you know, and it's frustrating for us as well sometimes, but, you know, my PhD started in 2002. And my project was to find this new gene. We knew where it was, we knew sort of thing, but we just couldn't quite nail it down. And it took us 7 years to do that. However, the technology that it took us to do my PhD took 4 years, and then it was 3 further years of work after that, you can now do in 4 weeks. Okay, so technology advancements, that for us is like the absolutely key thing, is technological advancements. And you know, and I'm sort of aware that all of this stuff changes, but you know, when I started, I worked in the field of ALS, MND, it was all I worked on, but we now know frontotemporal dementia is a really key part of the disease for us from a research point of view. And as Nigel says, like looking forward, we now work much more closely with the frontotemporal dementia researchers, because there's so much overlap between the genetics of the two diseases. So suddenly it's sort of brought in a whole other lot of researchers, which means we're talking to lots of different people, trying lots of different things. But it really is, for me, the technology that has allowed us to move forward. And one of the things that I could do, if you'd like, is I could share my screen and I could show you how our technology has allowed us to just look at a couple of different things. So Dean, do you think I could do that?
Do that, yeah.
Let me just find, you need to click on the button that says other than host can share screen. Well, I'm very used to this. I mean, for those of us who've, I mean, so I no longer am actually in the lab day to day. I only occasionally pop in, and I work on, I discovered, Nigel alluded to, I discovered this gene eventually that we published 7 years after my beginning of my PhD. I still work on that. That is the sole focus almost of my research, is this one gene. That's what I work on. And what I was going to show to you was, if I can get this up, hopefully you can see a motor neuron, or a pretty picture of a motor neuron. And this is what I work on. Okay, this is my my life is working on the insides of neurons, nerve cells. And in this picture, I don't know if you can see, but there is we have the cell here. This is actually in your spinal cord, and then it goes out from your spinal cord to your muscles. Okay, and they form these junctions. And I'm not going to go through this, but proteins travel up and down your cells, and they travel all the way down from your spinal cord basically out to your muscle. Proteins travel, and when proteins go wrong, that process is affected. And that's one of the things that I study. One of the things that the studentship that you are funding is going to be studying. And if we focus in on where this box is, and we focus in, this is where that nerve cell joins to the muscle. Okay, and that is a really, really key point, because that is one of the earliest things in motor neuron disease that happens, is that your nerve cells stop those connections with muscles. So one of the things that I work on is trying to work out why those connections stop. And from a completely different point of view from Nigel, he's very much sort of big picture, and I'm very much tiny molecular picture. Why do those nerve cells stop talking to those muscle cells? But if we focus in really on where that is, this is what it used to look like. You used to be able to take images of what they're called neuromuscular junctions, these connections at the terminals between nerve cells and muscles. And the protein that I work on is called FUS. And some of you may have heard of it because it's one of the genes for familial disease. And really, I just wanted to show you that we look at this, and I'm really sorry for anybody who's colorblind, because we always use red and green in microscopy, and I really should know better. My dad, my husband, and my son are all colorblind, red, green, and so I really should know better than this. But we always use red, green merging to make yellow. And technology advancements, we took this image probably 4 years ago. We submitted a paper, the reviewers asked us if we could try something to improve this image, and we got this image. And that image, a little bit blurry, you've got red and green, you can't really see that they don't overlap. If you aren't red-green colorblind, you'll be able to see on this image that the green actually sits within the red. Okay, so technological advancements have allowed us to actually distinguish right down, we're talking tiny, right at the end of your nerve on a muscle cell, allowed us to show which proteins are actually where. And they also allow us to just pick out further details. So if we now focus on the axon, so that's the bit of the nerve cell that goes from your spinal cord out to your muscle. So the nerves that go from that bit from your spinal cord out. And you may not be able to see these, but these are videos, and these are mitochondria. So these are your batteries of your cells, the little bits that you need for your energy, the bits that give you everything that you need to be able to move. And I have a wild type means a normal cell, and then I have 2 mutations of FUS. And the third one, the one that is called a truncation, is actually a video, but you would never know it, because basically that version of FUS is such a severe mutation that all of the mitochondria in that cell stop moving. And that, you know, that was a really key discovery for us, that when you have mutant FUS, that the mitochondria, these batteries, stop moving. Because if they're stopping moving, then that cell is not going to survive. So that tells us that there's a really key thing happening in those cells. And the studentship that started in October, that was funded by you guys, is that it really has allowed us to recruit an amazing PhD student called Sarah Taccanelli, who's Italian. Join my lab now, and she did her undergraduate at King's. She came top in the year. She got 3 prizes for best lab project, etc. So she's absolutely phenomenal student. She really is the future of research, as Nigel was talking about. She really will be brilliant. And she actually did both her BSc project, so her undergraduate project, and her master's project in my lab as well, which was very convenient, because when she started her PhD, she was able to start, because anybody who hadn't been in the lab before was not allowed to start, because COVID meant the shutdown of our labs for quite some time. So she's now this studentship will run for 3 years. It only started in October. And so we're gonna use more technologies, more novel, exciting technologies. I actually work on mouse models of ALS, MND. Like Nigel, I use the 2 interchangeably. And I will be able to use these mouse models to really look at the brain and spinal cord. As Nigel said, it's really hard to look at brain and spinal cord. That is our complication with almost all of our research, is the fact that we are working on a disease that is not easily accessible. You know, until we published the fact that this protein FUS that I work on was at the terminal where nerve and muscle interact, nobody knew that it was there. So it's quite hard to look for these things. And as I said, technology is improving all the time. And that's why I think that at the moment we've got lots of different groups. We've brought lots of different fields in recently. It's there's been an explosion of papers. It used to be that you'd get maybe 1-2 papers a day published in your field, and there's now, you know, I get 30-40 papers a day coming through on my emails, sort of, you know, that are of interest to me. So there is a lot more interest in the field, so that gives me good hope that we are progressing. And as I said, the technologies that we now have, particularly my interest, which is microscopy, which allows me to take pretty pictures, really kind of is going to allow us to really move the research forward. And as Nigel says, there's now a lot more interaction between the pathologists, the clinicians, the researchers, and this also helps us to move forward. So hopefully at some point in the future, I will be able to come back, hopefully in person, and sort of report to you on how that project has been going. But I really wanted to just say thank you very much. Hopefully you can see that, you know, even after however many years that I've now been in the field, I'm still fascinated by it, and it's still amazing. Things change all the time. Technology is, you know, brilliant the way that it has, we've adapted to it and it's adapted to us. So I'm not really gonna talk about anything else. Any questions, please ask. I just really wanted to make you see that, you know, things like technology are really what are allowing us to drive forward.
Can I just say, for me, that was incredibly interesting. The idea that I'm seeing the macro from Professor Leigh and the micro from you in terms of where things might be going, and that you're so positive about technology is a joy. I know that doesn't really help, but I think what we're getting is some form of positivity.
Yeah.
So I think for us as a branch to have you both here talking in the way that you have is great. We as a branch have just given £5,000 to your research, which I hope will help.
Yeah.
And now it'd be great if anybody has any questions for you. That it's quite technical, but if anybody has any questions, please ask.
The problem with being a molecular person is that you end up very sort of, it's quite hard to sort of step back from being inside that cell.
I have a question. So, Caroline, you intonated about the dementia, and obviously I've heard about Alzheimer's as well. So, obviously we know MND is, you know, it's perceived as quite a rare disease, and therefore doesn't attract the funding. And obviously we've all been excited by the coronavirus and the quick turnaround and all these things that one of the gentlemen mentioned earlier on. But I suppose the bigger question is, how does this connection with Alzheimer's and dementia, does it attract new investment? Does it attract new pharmaceutical interest? What's your view in terms of now these connections have been made of what the possibilities could be?
I think it does. So King's recently got what's called a dementia research initiative set up there, which is a centre in which there's a lot of funding. And although it's called the Dementia Research Initiative, almost all the people who work in that actually work on motor neuron disease. So it's because the protein that you may have heard about called TDP43, which you may have heard of, and there's a gene called C9orf72, which is the major genetic cause of both motor neuron disease and frontotemporal dementia. So because of these links, so in the field when I started 2002, there was one gene for ALS and there was one gene for frontotemporal dementia, and that was it. They were separate. 2006, the two fields came together. And following on from that, actually, yes, there's more initiative. Tapping into dementia funding, there is a lot more funding for dementia. Now, dementia also think they don't get any funding, obviously. Everybody thinks they don't get enough funding. I mean, that unfortunately is life. We all think we don't get enough funding. We often have looked at cancer funding going, oh, because cancer sometimes, like the Institute of Cancer Research, sometimes has more money than it knows what to do with. But, you know, they've really struggled this year as well. Everybody has struggled this year. And, but to be able to share some of the dementia funding, because it does get more, has been really, really good. It has, but it's also allowed collaborations between research groups who maybe previously wouldn't have collaborated because they would have considered themselves different diseases or different functions. And I would say one of the things that is good is that we are able to, with the power of technology like Zoom and things like that, you can have a meeting with somebody in America, you know, in New Zealand, wherever you're working with, and it's super, super easy now to have these research projects on a variety of things, like the same protein or the same cellular function. Like if you're interested just in that bit, as I showed, the middle of the axon, something like that, people's research will be focused just on that tiny bit of the cell, or just on the junction where it joins, or things like that. So I think there is more money. And I think there's more opportunity for shared resources, which is actually a key thing.
Amazing.
Alistair?
Dr. Vance, can you tell me if your project is now fully funded for the 3 years that it's supposed to run?
Yes, it's now fully funded, which is great.
Excellent.
Thank you.
Graham?
Graham?
Hi. Graham's only been diagnosed since June. We're still waiting to go on a clinical trial. And we read that it was rare. But since that time, I've spoken to and met so many people that have it. Do you think it should be still classed as rare, especially if that's what's holding back funding? Is it?
I don't know. Nigel, I don't know whether you want to jump in on this one. You're still muted, Nigel.
Nigel, you need to unmute. There we go.
Sorry. Yeah, no, it's a very important question. It is a rare disease. If you take 100,000 people every year, that's in the cast your net across the population, and really in 100,000 you get, in that year that you are watching them, you get, say, 2 patients with MND. So it is by definition what is the European Union and other countries define as an orphan disease. Now that actually has some benefits because it provides an incentive for drug companies to invest in it. Otherwise there wouldn't be a market, because it gives them a prolonged patent protection. But yes, I mean, it does have the drawback that it doesn't have the clout, say, of a panic factor of Alzheimer's disease, which gets straight to the hearts of politicians, doesn't it? On the other hand, what I think, and what Caroline has shown, is there's an opportunity value that I think we have at least an equal insight into disease mechanisms in MND as they do in Alzheimer's disease, through the work of people like Caroline herself and other people. And what struck me with Caroline's work, looking these beautiful, elegant techniques, what Caroline discovers is quite likely to be relevant what other people are discovering with other gene mutations. So there's an immediate, what I talked about, coming back to point the finger at the common mechanism or common mechanisms, which we can then target. And I think that every little bit of that jigsaw is vital. And it may be a very rare gene lesion. It may only be, perhaps 2 or 3% of all cases of MND contain that particular mutation, maybe less. To me, that doesn't matter. It's what I learned from Caroline's work about the mechanism in the cells that then I can translate to other experiments and to other genes, and that is happening all the time, and then take back to the clinic. And that is now happening at phenomenal speed, and as Caroline correctly pointed out, with collaborations with other people studying dementias and so on and so forth. So it's an amplifier effect. And the technology, as you say, Caroline, is transformational.
Anybody else? Judith.
Oops.
No, you were unmuted, Judith. You need to unmute now.
There, how's that? I'm not sure. I'm not sure who the question should be directed to, but I just wondered if there's been an increase in the percentage of MND running in a family.
No, I wouldn't say there is. There are. The C9orf72 mutation is the most common one, is a slightly strange mutation from a purely genetic point of view. It's a slightly strange mutation, and that may well have some certain factors that affect younger that will affect the future generations. But other than that, no. What I think is more aware is that clinicians in the field, sorry, Nigel, are more aware that you get genetic forms of the disease, so that they now understand, there's now a lot more understanding about the fact that you get genetic forms of the disease. And as I said, because it's so much easier now to screen the genetics of individuals, a patient comes into clinic at King's, we will take blood from them regardless of whether they have a family history or not. When they come in, we will get that blood, we will extract the DNA, we will sequence that DNA, and we will now compare it to our, databases. And that means that, but again, technology, something that previously you would have had to have individually gone through and done all these bits. We've now got, so Amar Al-Chalabi and Chris Shaw have got lots of funding to just sequence all the DNAs that come into the clinic, which means that all we're actually doing is just adding information in. Those people, there were always these people with this, but we didn't have the funding or the technology in which to actually sequence every single person. So I don't think there's more cases of genetic. I don't think it's increasing in families. I just think we're getting better at noticing it, if you get what I mean.
Okay. Thank you.
Can I comment there? And because I think it's a very important clinical. Caroline's absolutely right, but what you're not doing is feeding that information back to all the patients.
No, we can't.
Because this is why people often, why aren't you testing my genes? And of course, with gene therapy coming along, we haven't had chance. I mean, it is very exciting, and the King's labs are very well advanced in that. The problem is, if I just put your blood in the post and send it off for genetics, you may regret what you get, the result you get back. In other words, I as a clinician, and I'm sure Chris Shaw, would never do genetic testing without genetic counseling. It would be quite unethical. To do it for research with consent, which is what you're doing and what we always did at King's way back, is quite a different matter. But the two have to be separated in order to protect people with MND and their families from getting knowledge which cannot be unsaid. And of course has a huge impact for families. So yes, by all means, always discuss it. Let's have that discussion. And if it's the right thing to do for you, we would test it and give you the result. If it's part of research, that's different. But it's crucial that we do get that information about as many patients as we can, even though it's privileged, you know, anonymous in effect, from, unless you want the result, and then it's a different process. So I'm sorry to go, but it's a crucial distinction.
Can I ask a question on the back of that? So you found this in relatively recent times, Caroline, from what you were saying, the genetic connection on the familial side. So now that you've got more technology and the link with dementia, are you finding that at the genetic level that there may well be not necessarily a familial link in terms of, you know, MND that runs in the family, that one person could have dementia or Alzheimer's, but then the other person could result or sort of develop MND, and therefore there's a connection at that level? Is that something that's a possibility? Or
so in actual fact, the work I did for my PhD, it was many years ago, was the family I worked on was this huge Dutch kindred. I mean, and when I say huge, the Sib ship, which so there was some great grandparents, and they had 14 children. And then on average, they had the next generation each had 12 children. So the family tree went like, you know, you had to draw it as a circle, basically, to be able to get it onto a page. It was ridiculous. And in that family, we basically identified the region of DNA where we knew that there was going to be a mutation. And this has turned out to be the C9orf72 gene. And in that family and in other families with a C9orf72 mutation, this chromosome 9 mutation, you get patients with MND, patients with FTD, and then patients with MND FTD, where they actually get diagnosed with both. And not necessarily always one before the other. Sometimes get diagnosed with FTD, followed by the MND, and then the MND, followed by the FTD. As I said, this was a huge kindred. There weren't actually that many members. I think I had, out of that huge kindred, I think, because it went down after the generation where they had 12, I had 2 generations below that as well. I had DNA access to, for those younger 2 generations. And I think that there were 10 people affected, or 12 people affected. So actually in a huge family like that, it wasn't everybody who was affected. It was only a small number of them. And that genetic mutation, and one of the things that we're really interested in is, if you carry that mutation, why do sometimes you go on and get MND, and why do sometimes you go on and get FTD? And the difference really, I mean, is between in MND, it's the motor neurons that degenerate, and in FTD, it's the ones in the frontal and temporal lobes that degenerate, and we do not know why. That would be one of those questions that we would go, we'd love to know why, when you can have this mutation, some of you go on to get this and some of you go on to get that, because it's just really interesting it's such a complex disease to work on, compared to, as Nigel said, you know, with coronavirus, with COVID-19, there was one virus that caused it. So in terms of doing a vaccine, in terms of this, they just had that virus and they could go from there to work against it. With MND and with FTD, as I said, we just don't know, even sometimes where we know what the genetic mutation is, why that then results in disease.
Are there any more questions? Thank you. This has been incredible, and I agree with you. Technology has allowed all of us to come together tonight to listen to you guys, and personally, I think it's been really, really interesting. In any other way, and I would like to say that hopefully, if we have an AGM, that both of you could come in person and talk at an AGM. And so that everybody could question you at an AGM, because it's been incredibly interesting. Thank you both. You've been fantastic. Thank you.
Thank you very much for having me.
Thank you.
Moving on to lighter.
And carry on the great work.
Oh, thank you very much. It's a joy to have you both around, actually. And I meet you, Professor Leigh, very occasionally. And it's just great to know that you're both there and that you can answer questions. And I'm sure if anybody has any other questions, they could probably email, could they?
Yeah, certainly, yeah.
That's great. Thank you very much indeed.
Okay then, thank you. I'll leave you to get on if I may. Goodbye, everyone.
Bye-bye.
All the best.
Take care. Thank you.
Well, moving on to a lighter note, we have a raffle because it's Christmas. So Nikki, where is Nikki? I've lost Nikki. Hi, Nikki. Nikki, do you want to call the raffle?
Right.
Nikki, Dean?
Yes, we're here. Nikki.
Yes, Dean. Dean, I don't believe you gave everyone their number, did
I
you?
just suddenly realised I didn't give everybody a number when they came in.
Ouch.
I've been texting you like mad to say, give everyone a number.
This is where technology goes wrong.
Exactly. It's all a bit. So, without further ado, Dean will give you all a number.
Okay, let me do this quickly then.
Bear with us, everybody.
So there's two.
Wow.
There's two hampers. There's two hampers to be won. This is the first one.
Okay.
Tea and gin. I'm liking that, tea and gin.
And this is the second one.
And
there's also two bottles of wine, a bottle of red and a bottle of prosecco. And Maureen has also kindly made her husband donate an MOT.
Someone's going to want this.
An MOT and a carbon check.
Well, you might if you've got a car that's a knackered heat.
So don't all rush to get too excited. But I shall leave Dean to give you all a number. I'm not sure how we're going to assign all the numbers to all the people that we're supporting as well, Dean.
Well, there's another 20 on. There's 50 on here, and there's 1 to 70. What I'm going to do it from. So
okay.
I've just done everyone's screen grab, so I can see who's on who. So I can count across the screen. 1, 2, 3, 4, 5, 6, 5, 15, 25. I've got 50 on a screen, okay, and I've screen grabbed them all. Okay, so we're going to go from 1 to 70 or 1 to 80 and then do it from there, and I'll be able to tell you who's on screen and then who's not. So, how are we going to do that, Nikki?
I have no idea, Dean.
And I would suggest someone's going to randomly pick a number between 1 and 80.
Come on, Frankie, you can pull a number out.
Yeah. Frankie, you can pull a number anywhere between 1 and 80.
Okay, yeah, Frankie, go. 20.
20, okay.
Frankie, have you got a car? Because you've got an MOT and a carbon clean, mate.
No.
Okay, right. I'm going to turn around and say I'm going to remove the committee members from the first hamper, if I may. Could I ask you for a second number, please?
37.
Right. 5.
And one more.
One more.
That would be Chris Album's family.
Yeah,
where are they? Where have they gone? Where are they?
They're here.
There they are. You won, Chris.
Chris, you just won the hamper.
Yeah. Chris, would you prefer the MOT and the carbon clean?
Unmute yourself, Chris. You got Chris, unmute. No, we are unmuted.
Well, we're not.
We are.
Oh no, you're okay. We can hear you now.
What's the choice, Maureen?
No, I don't think anybody wants my MOT or a carbon clean.
No. I'll have the MOT.
I'll have the MOT.
Okay, fair enough. MOT gone. So we still have a hamper. So Chris, can you give me a number between 1 and 80, please?
I want to quite like the hamper. Hang on.
Which you want?
Okay, number 1.
No, you can't have that. No. Can you give me another number other than 1?
Number 2.
Oh, that's no good either. Right, carry on. Another one. I'm just removing.
Number 3.
That'll be Frankie.
Yeah.
I'll take a few more.
Frankie. Well done. Frankie and a family. There you get the hamper.
Oh, thank you.
Can we get another number from you, Frankie? One for 1 to 80, please.
15.
15. And that would be Dawn.
It's Dawn.
Like just waving here. There you go. There she's waving.
Brilliant. Well done, Dawn.
Well done.
Fantastic.
Dawn, could we have a number from you, please? Anywhere between 1 and 80.
69.
Okay, that's somebody who's not here, so we'll keep that one in reserve.
I think we're done now, Dean.
Is that right? Those things?
Yeah.
Okay,
Yeah.
lovely. So we're going to go to a quiz now.
No. We're going to go to thumbs up.
I thought there were some drinks going out.
No, that is so. Frankie's got a hamper. Dawn's got a hamper. Chris has got the MOT, and number 69 has got the wine.
Perfect. Fine.
Can we do thumbs up with Alistair now?
Yes, you can. Yes.
Thank you.
Alistair?
Yeah, I'm unmuted. Can you
Yeah.
hear me?
Mm-hmm.
Yeah.
This is the end of the Thumbs Up Club year. This is the last draw of the year, the Christmas draw, and we have 4 numbers to draw. The prizes are £30, £20, £15 and £10.
Alistair, can you explain to people who don't know what the Thumbs Club is quickly, just what it is?
Okay, the Thumbs Up Club is a regular monthly draw. People subscribe in January at the beginning of the year. The subscription is quite small. I can send forms to anybody who is interested for next year. Just drop me an email address and I'll send you the form. We then draw 2 numbers every month throughout the year, with the exception of Christmas when we draw 4 numbers, and the Christmas prizes are much bigger than any other month of the year. The other half, the prizes represent half of the money which comes in from subscriptions. The remaining half of the money goes to fund the projects of the branch. And because most people can gift aid, it's aided by gift aid. Whoops. Sorry about that. So, without more ado, Valerie, can you please draw the first tile?
It's 44.
44 wins the first prize. If anybody here has number 44, can they please shout? Second one coming up.
It's 5.
Number 5 gets the second prize.
And the 38 is the next one. And
Finally, 26 is the final prize. Nobody shouted, so the probability is that nobody is here tonight. Those people will all be notified by email tomorrow, and the prizes will be in their bank accounts next week.
Thank you, Alistair. That was great. Thank you. Now, if everybody wants to stay, Nikki is going to do a quiz.
Dean, actually, I've made the quiz, but Dean is going to present the quiz.
Well, I've just had on the on a text message.
You're going to win this fabulous prize.
Oh, there's what? Sorry.
There's this fabulous prize for the quiz.
Perfect. Well, also, is Dawn still with us?
Yep.
Yeah, she is. Did Dawn give us another? Oh, she did give us another number, didn't she? 69. Right. Okay. Fine. Right. Bill, I'm going to ask you if you would pick a number between 1 and 80. Furniture and Mirror in Chichester have just offered £50 off anything in store.
It's a gift voucher.
Yeah, it's a gift.
It's a gift voucher, Dean. 50 quid, not £50 off.
Okay. Gift voucher, £50 in their Furniture and Mirror store in Chichester.
Or Rustington, doesn't matter.
Lovely. So, Bill, in that case, would you mind picking a number between 1 and 80? If you're going to do it on screen, actually, do 1 and 50.
1 and 50, 13.
That's you, Mr. Alistair Sharp.
No! So are we now moving on to your quiz, Nikki?
We are.
Yes, hold on.
So the first one, you have to name
Okay, hold
the
on.
film, I'm thinking. I'm not sure what Dean's doing.
There we go. Can you see the screen? Oh, no, wrong screen, sorry.
There's 20 questions, and I'll ask at the end who's got all 20.
Do we have to write them down or call them out?
Blimey, Nikki, we'll be here all night.
No, you won't. You won't. They're just pictures.
Do we write them down or call them?
Write them down, and
There you
then tell me what films you see. And there should be 10 on the screen. Can we count? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Yeah, I want all 10 films, please. I'll give you a minute to look at them.
Yeah, that's long enough.
You got them all already, Maureen?
No. Who's the hot boy in the Santa hat at the top?
Snowman.
Can hear you.
Love, actually.
Hey,
You might want to mute everybody, Dean. They're all going to cheat otherwise.
I think this might be the Allborn family.
Taking it very seriously.
I was going to say, it's got
Oh, that's my favourite.
Hello, Bob.
Oh.
Benny, say it again.
Hello.
Well, I better write them down.
Lovely to see you.
Thank you. Lovely to see you too. Lovely evening.
Good.
Really good.
Oh, wow.
How many are there?
10.
10. I think I've got about 6 so far. I don't even know what they are.
Stu Kruger is taking this so seriously.
Oh, yeah, rather.
But it's a fantastic prize, Sue.
Yeah, what is it? I couldn't see it.
It's a Bayliss and Harding sort of, Christmassy bath thing.
I never get presents like that.
Yeah.
Lee, you're taking this seriously as well.
Have they got numbers on them, or is it?
No, but that's fine. We haven't got time for all that.
Just got to get 10 right, sort of. It doesn't matter what order.
No.
Don't know them. Mm.
Right, I've written them all down. Has everybody else finished?
I think that's long
No,
enough.
I can only get 8. I can only get 8 of them.
Does anybody need any more time?
No.
Yeah, not just you, Lynette. Anyone else?
No. I work on the basis that you either know them or you don't. It's not going to suddenly hit me.
Okay, round 2, you've got to name the Santa. Doesn't matter what film, just name the Santa. There should be 10 here, Dean. There's only 5.
There's one on another screen as well.
Okay, well, we'll let you look at this screen for a bit.
First.
Go back to the first screen.
Many of them.
When you say name the Santa, is it not Father Christmas? Or...
So you name the actor.
Oh, okay, right.
don't get the gorgeous one at the end, there's something wrong.
Is that your son, Nikki?
Yeah, that's my husband. In my dreams.
Looks like one of your sons.
Oh God, don't tell him that. It would go to his head.
Be the son of Helen Mirren. Let me move on to the other screen. All right.
Yeah, next screen.
2.
Don't know any of them.
I recognise him. I can't think of his name is.
You've had all day to look at them.
Is that one in the middle at the end? Taken.
Middle at the end?
Sorry, the last row in the middle. Is that the guy out of Taken?
No.
Oh, it's the guy out of 20.
Yeah, right.
Sorry.
Yeah, we need a bit of help. I don't know any of them.
Well, he's married to Goldie Hawn, if it's the one in the middle.
It is.
Oh.
It is. Okay, long enough?
Sure, it's the first one in case you didn't get them. Have you had another look?
Yeah. Right, who's go back to the big screen, Dean, so we can see everyone. No, not your screen, so we can see all the people.
Okay.
Try hard to get. Right, who got 20? Hands up, come on. Really?
Melbourne, probably.
No, you didn't. Okay. Anybody get 20 now? I can't see anyone. Can you see anyone saying who got 19?
Did anybody get over 15? Oh, Roberts, Arbery.
Wow.
Wow, okay, there's one there.
How many did you get, Robert?
Well, Maggie kind of put her hands up to 15 plus, but a couple of these could be guesses per se.
Right. Anybody more? Anybody else want to go? Anybody got more than 15 or more? Right. Let's see what Robert's got.
Well, I had 20 guesses.
Go on then. Give me the films first.
I can't. This take too long. I'll pass on that one.
Home Alone.
Well, do you want us to have a stab at 15?
Yeah, go on then.
Yeah.
We'll tell you what we've got.
Yeah.
Starting with the films.
Yep.
We've got Home Alone.
Yep.
Snowman.
Yep.
Elf.
Yep.
The Holiday.
Yep.
Muppets Christmas Carol.
Yep.
White Christmas.
Yep.
Love Actually.
Yep.
Die Hard.
Yep.
Great Christmas film, by the way.
Father Christmas.
Father Christmas.
No.
No. It's a Wonderful Life.
Yeah.
We got one wrong, that was all.
Yeah, one wrong. So the one you didn't get was that one, which is the Santa Claus.
Ah.
No.
Right, so that's a pretty good 9 out of 10.
That's very good. It's very good.
Right, let's the Santa ones. Right, what did you get on the Santa ones?
Oh, we had a go at that.
Yeah, we struggled with this one. Richard Attenborough.
Yeah.
Ron Moody.
No.
Oh.
That's not a surprise. That's
You've got 14 so far, yeah?
Tim Allen.
Yeah.
Ben Affleck.
Yeah.
Liam Neeson.
No.
That was the Taken thrown you.
Yeah, I threw that in there because someone was talking about it.
And Russell.
And Russell, but I can't think of his surname. I can't think of his surname.
Kurt Russell.
Kurt Russell, that's it.
So you've got 9, 10, 11, 12, 13. Does anyone get better than 13? Oh, I think the Carey's
I
think I got 17.
Oh, the hand now.
Yeah.
Oh, come on then. Come on. Come on, give me the Santas then.
Well, I got Jake Gyllenhaal, but can I get an extra point for pronouncing his name? It's Gyllenhaal. That's how you properly say his name. I'm a big fan.
My husband.
And also, I think some of the other ones he didn't get was Billy Bob Thornton and John Goodman.
No.
What about Ed?
Sorry, who Goodman did you say?
John Goodman.
Oh yeah, John Goodman, yeah.
What about Ed Asner and David Huddleston?
Ed Asner's one, not David Huddleston.
And for the missing film, was that the Santa Claus?
Yeah, the one with Tim Allen in, yeah.
Yeah.
So nobody got, go down to the next screen, Dean. Nobody got Leslie Nielsen. It's a picture with a little girl. And nobody got Jim.
Yes, we got Leslie Nielsen.
Did you?
And Jim Broadbent.
And we got Jim Broadbent.
Jim Broadbent. Yeah, that's right. That's right. So I don't know now. So who's ever got the most had this amazing prize.
So Becky, do you think that you beat the other person?
Well, I got 17.
Oh, I mean,
that.
Yeah. Okay, Becky. Goes to you, Becky.
After we gave them 13 of them. Who had 15 plus? Your hand didn't go up, did it?
I put my hand up and I did a little reaction, but I think I must have been on a different screen. I was doing this.
Good inquiry.
Yeah.
Hand up.
Technical
That's a thumbs up. That's not a hand up. Someone can't follow rules.
I'm actually a race steward, and I'm asking for a steward's inquiry.
You can have my prize if you want.
No.
Right.
All good fun. All good fun.
We take this beat well.
So back to you, Maureen.
Okay, fine. Not a lot to say now.
Oh,
Hopefully, I mean, I'm not joking. This night has just been such good fun. And I didn't think we'd ever be able to get something like this together. And I really hope that everybody has enjoyed themselves. It was great to have some fun and to have two really solid researchers allow us to ask questions and talk to us about what they do. We fund them, and I think that's fantastic that we do. And I hope everybody's really enjoyed it. I've had a really good time. Well done, Nikki and Dean. It's not an easy thing to organise. You've been amazing. So thank you very much. Honestly,
Well done.
absolutely fab.
Yeah.
Thank you. I don't know what else to say really. Oh, well, other than farewell. Thank you all very much.
So there's two things coming up. Next week, we've got this Saturday coming up. There's the Santas on the Seafront down in Worthing that Sue's running.
Yeah.
So we've got, I don't know how many people already signed up for running down on Worthing Seafront. It was like the beach December dip yesterday.
People have to do it in sixes because it's quite narrow. So there's people from the running club. I think people just come and they'll walk or they'll run, but there's quite a few people coming to support as well. So it's
Come
Can
down
I
to
just
stay
sign
morning.
up or can I just come along?
yeah. Oh yeah, it's fine.
Come along.
No, we're expecting you to run.
No, I will run, but what time is it? What time is it? And I'll come.
At the the splash point pads bit, that bit there, at 10 to 9, and then it starts. Part one starts.
And which day was this? Sorry.
On Saturday.
Perfect. Yeah, I'll be there.
Okay, cool.
So,
Going to be singing.
can I ask, where are you actually going to? As how far along? I mean, you're going east, are you, towards
yes,
Brighton?
Doing the route of the normal park run, but if you go onto the event that I think Dean's put on, all the instructions are on there, and then the route is actually on there, but it's the Worthing park run, but obviously they're not doing the park run at the moment.
And it's just going along the seafront, is it?
Okay, but on the way back, we might do a little sneaking up the pier just for good measures,
Yeah,
on the way back.
just past Grand Avenue goes up.
Yeah.
On Worthing Seafront, yeah.
Okay.
So all I can say is
We've also got the and just another thing is we've also got a another Christmas well, it's sort of a social group for the other Christmas that Sue's running. Sue, when's that going to be?
Yeah, that's next Monday afternoon at 3 o'clock, I think. It's our monthly tea and chat, very informal meeting, not as highly organized and brilliant as this one. Just total chaos, where we like, you know, everybody to have their say. And we've actually, these things sort of evolve according to the people who come. And we've actually got one lovely man who owns some alpacas, and he's going to show us a short film about his alpacas, which is just delightful. And you've given me an idea. I think we'll have to have another little Christmas quiz, because that was such fun. And yeah, and then we want people to say what they would like to do, what they would like to talk about. And in the future, there might be the possibility of breaking off into different groups, like a carers group and something like that. So please join us. This is a group that evolved from, we used to meet in a garden centre on a Monday afternoon, but obviously we're not bounded by borders now, so anybody can come from anywhere. We'd like to see people, and come along and you have to make yourself a cup of tea. If you don't like tea, you can drink coffee. Wine
3
is
o'clock, surely you can have a drink at 3 o'clock.
well, it is, we do frown upon it, you know, but people with us a drink. So.
No hope for you and I, Maureen.
No.
No, it's just a very informal, we call it a sort of tea and chat for anybody who's interested in and involved with the MND community. So we'd love to see you next Monday at 3. And Dean, I believe the sign-in is the same, isn't it?
It's the same sign-in.
Will you be there, Dean? Will you be signing us in?
Yeah, it'll all be there, and it's all on Facebook and the website as well. And yeah, and we're still taking lots of new ideas for fundraising for next year as well. We've already got Steve and his Tesco crew are doing one thing. Olivia Alban, she's got a whole running across the whole South Downs is going on.
Me and Chris.
So yeah, basically any way that we can do to try and help raise money and fund those amazing researchers will be amazing. So yeah, please, any ideas you have, we'd love to hear from you.
2021 is looking really good. We've got Judith is here, but we've got lots of events, and if we can run them all, it should be absolutely fantastic. So 2021 is looking really good.
Yeah.
And what is a joy is that more of us have met on Zoom, so we begin to know each other. I've really enjoyed it. I hope everybody else has.
Been a great night.
Yeah.
Good.
Great meeting. Yeah.
Thank you so
Fantastic.
much. Thank you.
Amazing, amazing.
Happy Christmas, everybody.
Happy Christmas.
Happy Christmas.
Happy New Year.
Merry Christmas.
Thank you very much. Thank you, Dean.
Thank you.
Okay.
Bye.
Bye.
Bye, everybody.
Yeah.
Bye. Bye, everybody.
Bye.
Bye.
Where's the Christmas music?